Thank you all very much for coming tonight. We're going to try and stay on time. Daniela, you can't go very far. We're going to actually do a pretty dynamic presentation and hand back and forth quite a bit between the two of us. And so tonight we're going to give a little bit of an overview about the Stem Cell Research Center, about UCI Health and where the center is going, where UCI Health at large is going, what that big hole in the ground is that's no longer a hole in the ground, but actually has stories next to it. And then we're going to put that in the context, what did I do?
No, you're good.
We're going to put that in the context of our research and how we've led, each of us independently, through a set of translational pathways, what it means to move from basic science into translational science, into clinical trial, and how that's kind of converged for us, and the great things about an academic medical center in terms of the collaborators that you have, how that's converged for us in the area of glioma and glioblastoma. So it's all going to be pretty high level. We're not going to make too many topic switches. You guys should be able to stay with us. So I'm Aileen Anderson. I'm director of the UCI Stem Cell Research Center. This is Dr. Daniela Bota, who wears many, many hats at the university. In addition to her basic science research program, she's also the director for the Alpha Clinic at UCI through the Stem Cell Research Center. And she's the vice dean for all of clinical research throughout the School of Medicine, which really touches on all aspects of the health enterprise. So we wanted to start out and give a little bit of context just about UCI and what an amazing institution UCI is. Again, this year, UCI has been named a top 10 public institution in the United States. I think it's easy to forget that for those of us who have UCI right in our backyard here, what an incredible place this is, how selective it is in terms of our applicants, and something that as a first-generation college student that I'm particularly proud of, that we do such an incredible job for diversity, equity, and inclusion. So more than half the undergraduate students at UCI are either first-generation students or underrepresented minorities. And I think this is a really incredible legacy that all of the UC, but particularly UCI, is contributing. And in that context, it doesn't stop with undergraduate education, but those efforts extend to our master's students and our PhD students as well. So here's to UCI. I hope that you recognize what an amazing thing we have right here in Irvine. With that, Daniela is going to tell you a little bit more about the College of Health Sciences, and then we're going to trade back and forth as we go.
So let me welcome all of you here. It's really humbling to see so many people. Some of them we know, some of them we don't. Some of them are students, some of them are physicians, some of them are researchers, and especially all our community friends. And I think it's important to talk about how much our College of Health Science has grown. And if you are coming around here and you remember when here there were horses and orange farms, and now you see all of those beautiful buildings, the first one that you could see here, it's our new Susan and Harry Samueli College of Health Science, built with the support of the Samueli family. And we have here amazing students. We have the School of Medicine, the School of Nursing, the School of Pharmaceutical Sciences, and our School of Public Health, currently still a program, but we are waiting for the announcement to be happening very soon. And why are we talking about this? Because in addition to our schools, we also have a number of institutes that are revolutionizing the way on which we practice, the way on which we try to give health and to restore health for the people in Orange County. So we have the Institute for Precision Health, the Center for Clinical Research, the Cancer Center, the Institute for Clinical and Translational Science, the Samuel Integrative Health Institute, and of course, the last but not the least, and actually the first in our opinion, the Sue and Bill Gross Stem Cell Research Center. And I can say this tonight, I'm not going to repeat it tomorrow, because tomorrow probably I'm going to talk about another institute, but moving to the next slide. This is the new UCI Health. So if you have received your care at any time at our hospital in Orange or our clinic here at Gottschalk, I will invite you to visit our new wonderful medical campus at the intersection of campus and Jamboree. And it is an amazing hospital. It's the first fully electric hospital in the United States. It's getting all kinds of architectural prizes for its own beauty. The Center for Advanced Care has opened at the end of April, And the Cancer Center has opened in July. So we are already practicing there. There is free and wonderful parking. You don't have to use the valet parking anymore. Please come to visit us. But more important, we have a new hospital opening in the late 2025. And that is a hospital developed in the interest of building neuroscience capacities, cancer capacities, and to support our research. Because if the hospital in Orange was hard to reach for our translational research, this we can go as a golf cart. More important, we have expanded UCI Health around Orange County, trying to serve all our patients, all our communities. And when I arrived to UCI 17 years ago, we had one hospital. Now we have a network of 6 hospitals crossing to the southern border of LA County and all the way toward Placentia Linda, toward Riverside County. So this is a testimony to our decisions to be able to serve all the patients from all the different communities in Orange County and above. The last but not the least, probably you had a little bit of problems parking because of this wonderful building that is getting built right now with the support from the Falling Leaf Foundation. This is one of the largest research buildings in the nation. It's going to have 215,000 square feet of basic and translational research. It's a medical innovation building where we'll all work together on special programs, again, to further human health. And the program that we want to talk about tonight is going to take place on the special area dedicated to the Stem Cell Research Center.
So we're super excited about this building because UCI, in the clinical sciences, translational sciences, the basic research in College of Health Sciences is really needed expansion space for a long time. Actually, the goal is to have a second building matching this one, taking up our other parking lot, so it'll be even farther to walk. We're going to have to provide golf carts maybe down the road, but that's fantastic growth for us to really match all of the potential that we have in terms of the work that's going on here. It was a competitive process for space in this building. I applied for the Stem Cell Research Center for expansion space, partnering in particular with Daniela, looking at a vision of really tackling neurological disease and injury as part of our expansion space there. Pleased to announce Daniela will be relocating from her current space into that building as our anchor. And we also have the opportunity for a number of new recruitments that's linked there. And really our goal here is to expand, the work that's going on now affiliated with the center that's really groundbreaking in terms of cellular medicine, cell and tissue engineering, and again, focused in this space that's about neurological disease and injury. Why is that important? Well, we're going to talk about glioma and glioblastoma tonight, but before we get there, I'm going to lead you down the path a little bit, saying a few words just about neurological disease and injury writ large. And one way that we can think about that in the field is through something that's called a DALY. So this is a disability adjusted life year. It just means it's the sum of the years of life lost either due to early mortality, you died before your time, or due to years that are lived with a disability. So my main area of research is spinal cord injury research. That's a perfect example of years lost due to a disability. If we look at that globally, for all the different types of conditions, including glioma, glioblastoma, where DALYs become a part of our calculus and thinking about what the burden of neurological disease is, it's 30 million years. 30 million years, which is really a staggering figure. That's autoimmune disease, cancer, everything you can imagine that touches on neurological disease and injury. And if we put that in perspective, 30 million years ago is when we first got the Himalayas. And 30 million people in a human chain would reach across the United States 3 times. It's a huge magnitude of impact, and it's why we think this is such an important area to think about addressing. So what then is the role of regenerative medicine and cell and gene therapies in thinking about this issue of neurological disease and injury? Well, obviously, stem cells are an important one. Stem cells are undifferentiated cells that generate all of the organs and tissues in our bodies. So this is how we grow during development. It's how some parts of our bodies heal themselves over time. If you have a cut on your skin, that's stem cells at work. If you have a muscle tear, that's stem cells at work, right? And the idea here is that we'd be looking towards harnessing that in the context of neurological disease and injury. And really, cell and gene therapies are creating this kind of next generation opportunity for new treatments for neurological disorders, and it's a very exciting area for us. At UCI, and indeed across certainly California with the California Institute of Regenerative Medicine, across the states, progress for neurological disease and injury, I think, is pretty well recognized that we need an integrative approach, and that's what we're focusing on. Here, that takes on three different pieces: discovery research, translational research, kind of the path in between, and then actually moving forward into clinical trials. We're going to tell you a little bit about what UCI is doing in each of these areas and then use our work as examples of these pieces. So before we quite do that, I want to make a point some of you in this room have heard me make before, but I think bears repeating as many times as I can possibly say it in front of an audience, and that is biomedical companies, biomedical treatments, either pharmaceutical companies, companies that are marketing drugs and new strategies, all trace their roots to academia, to academic research pipelines. There isn't an example of one that doesn't rely on academic research. I have said before in this kind of audience, there's nothing that makes me crazier than seeing those commercials for pharmaceutical companies where people in white coats are gleefully developing new cures that are going to treat patients. And it's fantastic, but they're bringing it the last step forward. Everything they're doing started in a building like this. And I think, again, it's really important to say that. Academic research drives innovation. If we look just in a one-year period, going back to the most recent paper I could find, over 13,000 patent applications were filed from academic research institutions like this one. This is the place that drives innovation that moves forward through companies. And in reality, if we look at a 5-year period, as has been published here, new drug approvals, every single one of the 216 new molecular entities that were brought forward in this period started with academic research and an academic patent. So what happens in buildings like this are absolutely critical for the process. In the Stem Cell Research Center, this is an example of what our current spin-off companies look like, right? So 6 spin-offs that have come out of faculty that are working in this physical structure, right? Their research driving forward to the initial stages of forming a new company, and hopefully will go forward into clinical trial and clinical application down the road. So what does that look like in terms of how we support it within the institution, this discovery to translational research to clinical research? Well, it's this building. It's the Falling Leaves Medical Innovation Building, as we just told you about. It's the Good Manufacturing Practice, GMP facility, which we've talked about previously at one of these public lectures. Over $13 million invested by the School of Medicine in main campus to build out this facility and create a path to be able to bring new cell and gene therapies forward. And it's also supporting the early stage clinical trial pipeline through the clinical trials that are run here by our clinician scientists. And Daniela is going to tell you a little bit more about that.
So when we think about clinical trials in cell and gene therapy in California, we are very fortunate because the California Institute for Regenerative Medicine has funded a number of 9 clinics. And those 9 clinics are clinical trial units where the treatments coming, the potential treatments coming from the laboratories go into human beings, many times for the first time for that specific approach. So you can imagine that in order to have the talent and the know-how to be the first clinic administering a certain experimental treatment to patients, most of the time the patients having very severe and complex diseases like we're talking tonight about glioblastoma, you have to have extremely specialized centers. So that's 9 of us. You recognize our 5 UC schools. UCI is one of them. And then Cedars-Sinai, Stanford, and USC. This is a network that has enrolled more than 1,000 participants in more than 200 clinical trials for more than 40 diseases. And the investment from CERM in different ways has reached $80 million. So, very important. This is where the treatments of tomorrow are getting tested. We had the opportunity at UCI to participate in excellent clinical trials which led to FDA approval for treatments from burns to hematologic disorders. Next. So our latest funding, which is an $8 million grant, we received it about a year and a half ago, established UCI not only as an alpha clinic, this was a refunding, we were an alpha clinic before on the first reiteration, but also as the hub of a center of a number of other hospitals in our community that we are trying to support to develop clinical trials and to offer the patients from remote communities, such as in Inland Empire, the ability to have access to those very innovative treatments, because it's very hard for the patients that again have severe neurological disorders to have to travel from Salton Sea or from Colton and to come to UCI. So this is our effort to span 4 California counties and to serve all age groups, as we have the Children's Hospital of Orange County, the Long Beach VA, as well as the multiple groups in Riverside. So when you look at the list, you can now see how we have extended all across those 4 counties. So what do? We do about everything that it takes to work with our academic investigators as well as with industry partners, biotech, pharma, to bring those treatments to our patients. So we do things from agreements, funding, writing projects, but most important, we have the research coordinators, the spaces, the nurses, the support staff to make sure that we help the physicians administering those treatments. We educate the physicians administering those treatments. And the patients receive the best potential experimental treatment, which sometimes is the last treatment that they could have for their disease in a safe environment, patient-centric. So,
okay, so that's our pipeline, right? Very discovery research and the kinds of facilities we're standing in, this idea of enabling the generation, the development of new cell and gene therapies, and then finally into how do we test those in humans, in people that have diseases, that have injuries in an ethically and clinically responsible kind of way. So with that large background, I'm going to tell you a little bit about one journey for this very quickly. That's from my lab and Dr. Cummings's lab, really focusing on spinal cord injury. And the reason I'm going to start there is to show you how discovery science can work in kind of twisty ways. And so I want to give you a little bit of an overview here. I first joined UCI in 2001 as an assistant professor. I focused on spinal cord injury and understanding the immune system in spinal cord injury, what went wrong, what might be good and what might be bad, what we could do about it. From a first serendipity point of view, we developed a kind of unique set of models that no one else was working with at the time. I was not a stem cell biologist, but stem cell biologists came to me in the early stages of neural stem cell work and said, we think that we could test our neural stem cells in your model because no one was using what are called constitutively immunodeficient animal models, a place where we could transplant human cells and assess what their potential was for repair. Not a stem cell biologist at that time. But nonetheless, we launched down that path. In fact, within 8 years, the work, all of the preclinical work that we did here went on to clinical trial for human thoracic spinal cord injury. In fact, it was pretty exciting at that time. We had a majority of subjects in that trial that had some improvements, at least in sensation, some in terms of overall level of function assessed on something called the Asia score. Very exciting at that moment in time. It was really one of the first big advances in terms of thinking about spinal cord injury. We consolidated that work over the next couple of years, looking for ways to treat patients who had had the injury for longer, treat people not just with thoracic injuries, but cervical injuries, and move forward, partnering very closely with the company that we worked with in terms of developing these cells. Around 2017, that came to a screeching halt. Why? Because companies sometimes have challenges in terms of how they move things forward. This is the bridge part that can be hard, moving out of academia and onto something that has to be supported by a company in order to move on to the later stages of clinical trials. We regrouped in my lab and said, did we do all that we can do because we did the basic in the preclinical science, or can we actually think about driving this forward to the next step? And we made the decision to really double down on that translational space. Katja Pilty, who's here in the audience tonight, wave your hand, Katja, dug in with no grant support, with philanthropic support that we had from the Johnson family to make our own cell lines, to make not just clinically compliant cell lines, but GMP compliant cell lines that could be used in future clinical trials. And we've spent the better part of the last 5 years testing those and trying to move them forward. And that gives you a sense of sort of what the timeline is that's involved in discovery science to translational science on into the clinical testing of the things that we develop. We started in 2002, and here we are in 2024, maybe just able to think about moving forward back into the clinic with what we've worked on for all this time. I want to give you a sense of what that looks like in terms of studies, because that process is a long one. These are all the different sorts of things that we thought were important to test, and certainly that the Food and Drug Administration felt was important for us to test to be able to move forward into phase 1 and 2 clinical trials. It's a lot of work that has to happen behind the scenes before this ever goes to a company. And then the last piece that I want to highlight is the sources of funding for this are really critical. At some early stages, all funding from the federal government. At in-between stages, a mix of venture capital and federal government. But really, the sustaining part of innovation over the last few years has been all philanthropic in terms of our work, and I want to acknowledge that. So along that path, we're understanding how these cells behave, how they function, and how we can harness that, thinking about neurological disease and injury. So one of the things that we started to understand early on working with neural stem cells is that the niche is quite important. All stem cells occupy a niche. It's just a place in the environment that those cells reside. And that niche can vary or change, right? If you think about neurological disease and injury, it can be in a state of repair or disrepair. And therefore, the cells that are in that niche are getting different signals at different times. A developmental niche, when you're making a new organ and tissue, isn't the same as one that is in the adult brain or the adult spinal cord. And it's not the same as one that's present if you have multiple sclerosis or Alzheimer's disease or spinal cord injury or cancer for that matter. And so, really critically at that basic science, the discovery science stage, is that understanding why something happens, in this case, why stem cells behave in specific ways, is important for our general knowledge base. But it turns out to be really important in terms of clinical translation as well. And this is where our story starts to bridge back over into the area of glioma and glioblastoma. So we discovered along this path, right, this 22-year path that I've shown you, that immune cells and immune molecules communicate to neural stem cells that we transplant. And that's actually really important. The cells that we transplant therapeutically are listening to that niche. They're listening to the environment that's around them. And one of the things that we started to do was investigate how that was happening and what the molecular signals were that were conveying those messages. And it turns out that there's two molecules that we think are really important, both a part of the immune system. One is called C1q and one is called C3a. I'm not going to tell you more than that tonight. But as we started to think about those, we investigated what they were controlling. And one of the things that we realized is that C3a and its receptor, C3a receptor, which is expressed by neural stem cells, is really important as a survival signal. So neural stem cells make this molecule for themselves. They talk to themselves with it in what's called an autocrine pathway, and that stimulates a survival response. They can also take C3A in from the surrounding environment. That's called a paracrine pathway, but they're listening to it all the time. And if you take it away, those neural stem cells die. Why would that be important? So we turned our heads to try and think about where that would be important in biology writ large. And one of the things that we came to realize is that beyond neural stem cells, cancer cells seem to use this signaling pathway. So this is an example. It's the only complicated slide I'm going to show you, looking at C3A and C3A receptor in glioblastoma and glioma stem cells, and showing that the expression of these two proteins that we became interested in is massively upregulated. And that suggested to us that in these cancer cells, that same survival pathway might be co-opted or even amplified. And that's when we went to talk to Daniela.
So while Aileen was having all this fun looking at her neural stem cells, science was moving also forward on the idea that tumors have also their own stem cells. So somewhere when I was a fellow at Duke doing my fellowship/postdoc, it has become apparent that glioblastoma has glioblastoma stem cells, which are very, very closely related to the normal neural stem cells to the level that they have common ancestry. This didn't come as a surprise because when we look together in the lab, and I have a number of my lab scientists here, to the plates that grow those type of cells, it is very hard to recognize which is which. They look immensely similar. Actually, that's how we purify the tumor stem cells, but making them grow the same way on which we grow the neural stem cells, in spheroids. They grow in the same media, they can form little bubbles, they can grow and continue to form differentiated cells. You have a stem cell and a daughter cell, and the daughter cell can continue to differentiate. And actually, we learn more and more that the brain tumors are trying not only to integrate and infiltrate the normal brain, but also reproduce a lot of the developmental pathways that we see with the normal brain. So brain tumors can form cells that look like astrocytes, they can form cells that look almost like neurons, they can form cells that look almost like the coating cell, like oligodendrocytes. So all of those things are characteristic for both type of tumors. Where the characteristics are different is where we test if those cells implanted in an appropriate niche can form tumors. And if you implant glioma stem cells in the brain of animals that are immunosuppressed, they will always form tumors. That's how we declare them successful cultures. The neural stem cells, by definition, should never form tumors when they're injected in the appropriate niche. There are some potential reports that when, unfortunately, unscrupulous researchers have treated patients with cells that are not fully purified and not on the appropriate niche, some of the so-called neural stem cells might have formed tumors. That is a scary story. That is for another night, maybe the Halloween. Message being, if you want to ever have a stem cell treatment or anybody from your family wants one, please come to one of our alpha clinics. But moving on, back to what we do in the lab and in my clinic, we try to do two things. We try to develop better treatments in the lab, working on patient-derived cells and patient-derived models. And then we try to bring that information in the clinic with two goals. And those are the goals that we always try to keep in balance. We want to increase survival, but in the same time, we have to maintain the patient's quality of life. And when you think about what I told you, this is where the biology becomes very, very important, because we spend in our research group our time trying to understand how to target those very close cousins in a separate way. Be able to eradicate the glioblastoma stem cell, but keep the neural niche and the neural stem cells as much as we can intact. So why glioblastoma? Glioblastoma, as many of you have heard, it's a very aggressive tumor. It is the most aggressive brain cancer. It's one of the most aggressive cancers in general. It is still a relatively rare disease. When we don't have a treatment for this tumor, it can double in size every 2 weeks. Unfortunately, a tumor that grows so fast can kill somebody without treatment in less than 3 months. But even with the best treatments that we have right now, in average, if we look at statistics, the survival is still less than 2 years. Why is that? Unfortunately, this slide, it's the history of my career and many of my colleagues' career. And what it's telling you is that we have done surgery since the 1800s. Chemotherapy, like the mustard agent, started to be approved with minimal efficacy in the late 70s, so was brain radiation. And since the 2005, 2000, and 2009, we have not had any drugs approved for the treatment of glioblastoma. In 2011 and 2016, an electrical device was approved that is generating electrical currents to control the tumor. All through this, we had almost 40 clinical trials, phase 3 clinical trials, that have failed to improve survival. Why is that? Why can't we get forward when we are getting so much better in controlling cancer in many other diseases like melanoma, like lung cancer, like breast cancer? The problem with the brain tumors is that they are in the brain. That's the biggest problem. And why is that a problem? Because we have developmentally developed a structure that's called the blood-brain barrier. And the blood-brain barrier, it's why many poisons that we can have, and maybe they're gonna give us diarrhea, they don't cross in the brain, and they don't infect the brain, they don't give us toxic syndromes. But that barrier also limits the delivery of cancer drugs in the brain and many other molecules. So with that, even if a drug gets to some level in the brain, many times doesn't form normal levels in the brain tumor, because the brain tumor has mechanisms to extrude, to push the drugs outside of the cells, which are other mechanisms of resistance. And then we already talked about this very fine balance between the cancer and the toxicity to the normal brain, and trying to achieve basically the grail, which is being able to get a drug in the brain, get a drug in the tumor, and only in the tumor, and in the tumor eradicate the most aggressive part, which are the tumor stem cells. So, this is again, as Aileen was trying to tell you, a story that has started in patients. When I came at UCI in 2007 as an assistant professor, very much like Aileen, I was very interested on trying to find drugs that were able to cross the blood-brain barrier. And talking to other scientists that are working on other cancers, I learned about a specific drug that is named marizumab, that was tried in patients with multiple myeloma and was giving the patients with multiple myeloma good cancer control in clinical trials, but also was giving them some confusion, some imbalance, suggesting that the drug can cross into the brain. So we took the drug with philanthropic support from the Stern family into animal models, and we were able to look at cells. And you could see there my beautiful stem cells, tumor stem cells, forming bubbles. And then we injected them on mice and created avatar mice. And if we let those tumors in avatar mice, they form very big tumors and we don't have more mice. But if we gave the mice, and when we gave the mice the drug, the marizumab, we realized that the tumors were getting smaller. And this is a very simple story because the story is much more complex. Of course, it took us a lot of work looking at things that Aileen described, safety, toxicity, different models. But we ended up with the FDA allowing us, and the company that was supporting us at that time, to go on 4 human clinical trials. And this drug was tried in many patients, almost 1,500 patients. And not only that, but it gave us the lessons to develop new molecules that you can see there in the corner, which hopefully we're gonna move things forward. Moving to the next slide. One of the things that's important for me is that even when drugs don't work for all the patients every time, they tend to work for some of the patients on clinical trials. And many papers have published in many cancers that participating on clinical trials, besides being something immensely generous of the patients, it's also potentially benefiting the patient. And this is one of our patients that has agreed with me talking about her story. And this young woman was diagnosed in 2017. And at that time, we, our clinical group, my colleagues and me, treated her with this drug, which targeted glioma stem cells. She has completed her treatment in 2021, and she's still alive. This is something that gives us the hope to continue to develop this type of drugs, to try to find ways on which more patients can benefit. But moving to the next thing, I know that Aileen talked to you about the niche. This is not as beautiful as the niches that she showed you. However, this is what I have to do when we think about the glioblastoma niche, which we're also calling the tumor microenvironment. And the problem with glioblastoma is that glioblastoma, it's a cold tumor. This goes back to evolution. We talked about, the drugs not crossing the blood-brain barrier, but also think about the other thing. If you have an inflammation on the brain, you don't want to form like a big infection, big abscess, because there is no space. What you want is that infection to be slow and smoldering, so hopefully the body can solve it and you can survive. Unfortunately, the other part of it is that the brain, in a way, it's more protected. It's a more protected environment against immune responses. As compared with what we talk melanoma, which is a very hot tumor. Blood can go there, immune cells can go there. So what we are trying to do many times in our research is to transform our very cold tumor into a hot tumor. And how you do that? You try to educate cells to go there and teach your immune cells to attack the tumor. So the dendritic cells, or DCs, are the cells that present antigens. And in the glioblastoma, they don't train. They don't leave from the lymph nodes, and they stay there, and they don't attack the tumor. But if you take those cells out from the patient body, and you teach them in the plate, in the dish, subject them to the tumor antigens, then you can get activated dendritic cells, and activated dendritic cell can activate your lymphocytes, your T cells, which are the cells that fight the tumor. Simple. We are just taking one episode from the body where it doesn't happen, and we do it in the dish. So, actually, we just did that with the support of one of the companies, local companies in Irvine. Serendipitously being a company that before was involved also in work on the spinal cord injury. We all take ideas from the others. Stem cells are stem cells. So what we did, we tried to purify the glioblastoma stem cells from the patients, from the surgery. Then we took cells from the patient's blood, we put them together in the dish, we obtained those activated immune cells and injected them back in the patients together with the standard treatment for this disease. So again, we have administered this treatment to about 60 patients. It's a subcutaneous, very simple injection. And what we have found is that the patients that got the treatment, their tumor was stable 50% more time than the patients that didn't get the treatment. We are still working on a phase 3 study. It is approved, but it's still fundraising. Clinical trials are extremely expensive. But going back to what I said, it's important to see the patients are doing well. So now the last thing that I want to tell you about is looking back at the niche, what if we are playing even more? So we started with the dendritic cells that are not trained. Then we have those cells that are called MDSCs that also are a problem because they inhibit the immune response. Pushing buttons. So now, if we are getting all of those cells together, we can create a better activation. So how do you create that? When you have surgery, you have the ability, after you remove the tumor, to inject in the tumor genetic therapies. And you could have those genetic therapies carried by viral vectors, and you can increase the immune system in the tumor while with one virus, while with the other one, you can start breaking the cells exactly like you'll have if you have an infection. And if you're also administering treatments to decrease those inhibitory cells, what you see is that red line where animals live better and better, longer and longer if you do that. So, with help from philanthropic support from the Cancer Center and the Anti-Cancer Campaign, we are opening a clinical trial right now to do exactly that in our patients, trying to see if by doing immune therapy and gene therapy, we can improve survival. So we are going back to our stem cells.
So, in Daniela's journey, you can see the influence of basic science through an understanding how the immune cells, that the fact that there are glioma stem cells that are very much like neural stem cells, and putting those together in the context of developing new therapies. How does that apply to what I talked about, our basic science discovery in terms of how neural stem cells work and how they're listening to immune cues in the niche? Well, we tested whether this molecule we identified, this signaling that happens between the immune niche, the microenvironment, and neural stem cells had an impact in terms of glioma cell and glioblastoma cell survival and self-renewal. And in fact, that was very true, both in vitro and in vivo. So in a dish on the left side here, you can see that treatment with a molecule that can disrupt that signaling interaction actually kills almost all of the glioma cells that are present in a dish. And if we look in one in vivo model of tumor initiation, in fact, by treating with this same molecule and disrupting how that molecule, treating with this same treatment, and anti-C3A can disrupt C3A and C3A receptor signaling. In fact, the blue line shows that all of the animals in this model were protected from a glioblastoma initiated in this animal model. So that's pretty exciting as a new potential path. It's not the only path forward. We have additional data that I think we're pretty excited about, that this pathway may be particularly active in that glioma stem cell population. So just like neural stem cells is where we identified it, the glioma stem cell population, which can escape other sorts of therapies for glioblastoma, is the one that seems to be particularly targeted by this particular approach. So we hope that this is a new path forward.
So we hope that we convince you tonight that our ability to develop better treatment for brain cancer requires the innovative approaches that move back and forth between the bench and the clinic and back to our benches. And hopefully together we can champion better treatments for glioblastoma in partnership with our CERM, NIH, academic institution, industry partners. But also I hope that you heard us saying that a lot of this work, before being supported by the federal funding, the state funding, or the industry funding, was supported and continued because of grateful donors.
And I would just echo that again to bring two things home, that definitely the work that we did with glioblastoma wouldn't have been possible without philanthropy. I only just got awarded last week our first glioblastoma grants from CERM to continue this work on in terms of development, and that actually is a collaborative grant between Dr. Bota and myself. So again, I would be remiss if I didn't bring this home by closing the idea that discovery research and translational research and clinical research are all one big interactive pipeline, that this is the heart and soul of what we do here in the Stem Cell Research Center from a wide variety of areas, not just what we told you about tonight, organ and tissue engineering, stem cell bioprinting, molecule screening for other types of diseases like schizophrenia or major depression, traumatic brain injury, Alzheimer's disease, Huntington's disease, Parkinson's disease, ALS. You get the picture. This type of translational pipeline is something that's about being at an academic medical center, and I think it's something that we're super proud of in the Stem Cell Research Center here. So with that, that is our last slide. Thank you for your attention, and we will answer whatever questions you might have.
First of all, I just want to say thank you. That was an amazing presentation. And so when looking for an industry that I really wanted to work in, I thought of medicine. And doing some research, I discovered this law called Irum's Law. I don't know if you guys have heard of it, but it's basically says that medicine gets harder and more expensive over time. And I think Dr. Bota also said this in her presentation that it has been getting harder. Like the last time you guys, or you had a slide that said like the last time you had something was in 2017. So yeah, we can see that it's getting very difficult. But along with Irum's law, there's something called Moore's law, which says that the amount of transistors on a circuit board increases like twice over a couple of years. So my question is, and a lot of this this research in like transistors, it goes into like machine learning. And my question is, are you guys integrating machine learning into making your research faster and really accelerating this pipeline? And if not, do you guys plan to integrate machine learning?
That's such a great question. So I have some comments to make too.
And I have some comments as well,
Please
maybe. So let's start with the importance of machine learning in glioblastoma and in medicine. We do use already algorithms and we already use machine learning. And what you do with that are real life data and real life clinical trials. So if one of my pet peeves was the FDA, and we don't have enough time to discuss my pet peeves with the FDAs, because there are multiple, is that any time that we run a clinical trial, we are asked to do a phase 3 clinical trials, which means that half of the patients have to be randomized to the standard of care. My question always remains, when we have treated patients with this disease and we haven't changed the prognosis for the last 20 years, why can't we use our large, immense databases like EPIC, which now covers all the patients in the 5 UCs, to generate and match patients with the patients in our clinical trials? So half of the patients don't have to receive a treatment that we already know is not achieving the best results. We have also published data on machine learning, which we can use to read MRIs. And just by reading the MRIs, develop numbers that show us what kind of characteristic molecular gene mutations tumor have. So we are working also with researchers in campus to try to build very complex mathematical modelings, because glioblastoma usually have specific mutations for specific patients, and there are thousands of mutations, and trying to understand based on those complex mathematical modeling, which pathways should we best target in order to achieve response. So there are a lot of activities, and I think it's going to be a very important component of our work.
Yeah, so I agree with all of that, and I would add maybe a couple of things. So it dips into the area of precision health, right? And that's where machine learning and AI and what Daniela was just talking about really come together, that as we're able to process the information from MRIs, from a high throughput genetic sequencing, from all of these different avenues of information gathering that have actually speeded up, using machine learning and algorithms that come from that are leading towards a precision medicine approach. So we have a better predictive capacity to say, this person we should stratify to this treatment, they're most likely to respond. This person B isn't going to respond to that. We should try something innovative and new, or go down a path where we're looking at whatever, a checkpoint inhibitor combined with something else, because based on their mutational profile, we predict that that will be the best response for that individual. It takes an enormous amount of data to feed in. The second law that you cited really comes to some technological advances that we've had. For example, the rate at which we can complete sequencing of the genome to be able to enable that, right, has been an exponential growth curve that really is directly linked to the transistor law that you cited, right? When the Human Genome Project was funded by the National Institutes of Health going back into the 80s, it took 10 years to complete the first full sequence of the human genome. Now we can do that in less than a day, right? So as technology advances, it opens up these different areas in the combination with machine learning. But the other thing I would just comment on quickly is that this is where, again, the academic enterprise and this basic science to translation to clinical pipeline become really important. I don't think it is the case that for at least the foreseeable future, like in your remaining years on this planet, that it is the case that medicine is only going to become more expensive. I think that much of the technology that we're developing now, because it's personalized, because we're moving towards cell and gene therapies that can be executed in an academic environment, we may actually bring the precision of treatment down and the cost of that treatment down with it. If we can customize iPSCs, if we can customize dendritic therapies and not have to go out to the early stages of technology where we are, where we're talking about hundreds of thousands of dollars of dollars to be able to treat a single patient, but bring that into something that can be done in an enclosed automated system within our GMP facility, we can probably get that therapy down to tens of thousands of dollars, and the impact of that is going to be enormous. Sorry, those were two long-winded answers.
I just have questions about like the basic science, if you want to. Okay, cool. So in the cold tumor, warm tumor thing that you guys showed, you said that you prime the dendritic cells to train the T cells. How do you prevent autoimmunity in that case?
The holy grail, as I mentioned, is to change from a the cold to a hot tumor. I do agree that autoimmunity in the brain would be something very bad. Unfortunately, we are yet to be able to train our immune cells so good to create an autoimmune response in the brain. However, it is also a matter of being able to purify the appropriate tumor antigens. So a lot of the work that we do is, all the work on dendritic cell vaccines took the whole tumor, the whole mass. There were blood vessels, normal neurons, abnormal neurons, and tumor cells. And those studies gave us a lot of ideas, still didn't develop autoimmunity, but gave us the idea, which I was showing you there, to create very purified glioblastoma stem cells and use only those to prepare the antigens. So that's the best way to avoid autoimmunity.
And I can add on to that a little bit. If you think about the one data slide that I showed with C3a receptor expression being upregulated by hundreds of folds in glioma stem cells, right? That's what you're betting on in that approach, that you're going to be training dendritic cells and T cells to be activated against proteins that are expressed on the tumor surface that are maybe not entirely unique in the body, but massively overrepresented in those cells specifically. It's the same basis on which we build CAR T cells, right, in terms of the antigens that we're asking those CAR Ts to respond to.
Sort of follow-up question is, one of your treatments is the anti-C3A. Yeah, C3A. Do you ever see an adaptation by the tumor to downregulate C3A and still survive?
So this is a challenge in models because
Can you get this guy's name and hire him?
it's a great question. It's not a question we can easily ask in animal models of glioma and glioblastoma. It's the kind of question that's going to come out when eventually, hopefully, we move forward into a phase 1/2 clinical trial, right? Asking how a tumor adapts is something we can get at a little bit in a dish, but in an animal model, those models progress very, very quickly, and it's difficult to look at tumor adaptations that are happening. The last thing I'll just say here is, in a dish, we kill all the glioma stem cells. It may be, right, so my dream result would be that that's just so efficient that and it's such a fundamental signaling pathway for these cells that they won't have a way to escape it. That would be really unique, but it's the potentially exciting piece for that.
I would also add that, yes, we have tried to block pathway by pathway, and some pathways are more effective than others. But the treatments that we have approved for glioblastoma improve survival somewhere between 2 to 4 months. So even if you don't eradicate the tumor, if you can develop a treatment that's safe and effective, and it increases survival with 4 or 6 months, that's already one step ahead. And in many cancers, the way in which we treat them, we have first line, second line, third line, even fourth line treatments. Because cancer, in the end, it's a chronic disease. We don't cure cancer, we control cancer.
Okay, and then one more question.
One more.
One more. Okay, so I was under the impression, forgive me if I'm wrong, that the complement proteins are just like floating around and they attach whenever possible or whatever. But how do these immune cells give the complement proteins?
Yeah.
And like, how do they communicate?
So also a great question, which I've actually talked on before at the community lecture series at one point. Let's go way back in time. The complement system is 450 million years old. It first shows up in horseshoe crabs. Horseshoe crabs don't really have a functioning immune system. They don't have a functioning complement cascade. So one of the challenges that we have in biology is that we give things names based on kind of when we discover them and the role that we discover them in. I think a lot of our data in the lab suggests that there are complement components like C1q and C3 that can function absolutely independently of the immune system. They had a job before we put together the immune system. It's just that they were discovered as a part of the innate immune system and a part of something that we called a complement cascade. And so that becomes the lens that we look at them through, right? It doesn't mean that's all they do. And in fact, in the central nervous system, in the brain and spinal cord, the early parts of what we think of as the complement cascade, those early molecules have clearly independent jobs that have nothing to do. So a paracrine way, so from another cell to a cancer cell or to a neural stem cell, it could be that they're getting a signal that comes from the immune system. That's how we identified their role, right? But in fact, neural stem cells and other cells in the brain and spinal cord use those molecules independent of anything to do with the immune system.
Thank you.
And that it's also highlighting the fact that, as Dr. Anderson showed, the treatment that she's trying to develop is going to be administered locally.
Yeah.
So this is one of the advantages when you think about things that might create systemic toxicity. You always have the ability to administer them locally in order to limit toxicity. So time will say what's the important systemically versus what's the important in the tumor microenvironment, but those are the type of questions that we have to answer before we can go to clinic.
Somebody new. Sorry, I'm going to diss you for a second.
Hi, thanks for an excellent presentation. My question deals with the item that you showed of the treatments that were approved recently, use of electrical or electromagnetic spectrum. Question arises, to these treatments are available for use or combined with the proteins, and how would they, and if they are, how do they affect, what's the difference in efficiency and efficacy? And the other question, slightly different area, deals with the factors that impact, as when you described how the receptors are showing up, the external factors that impact that some people respond and some do not.
So I will start with alternating electrical fields. The technology is called Novo TTF or Optune. The general idea is that if you put a tumor in an electrical current, the tumor are forming dipodes, plus and minus. They're trying to alternate the electrical current, moving right to left. You are putting stress on the mechanism the cells use to divide, the mitotic spindle, and the cells break. Interesting enough, of course, this technology was tried first in cell systems. It was tried also in mice. We do have the special system in our lab. And when we look at other experimental therapies, one of the things that we do, at least in the dish, is we try to combine them. And we are finding many times that the combination is more effective than either treatment alone. And that's why we do a lot of synergy experiments. So that's how we do that. So we don't know yet in mice. It would be kind of hard to put a little electrode on the mice head and they to run around. But we always test that in the dish. In parallel, when we go to clinical trials, if we go to where this device is approved, which is newly diagnosed disease, we never exclude that from the way that we treat the patients, because if something works, it's unethical not to offer it. So I will go back to the receptors, to Dr. Anderson.
So the question, let's come back to that question. That is what may upregulate them or modulate them. Yeah. And yeah, so it's a really interesting one. Actually, particularly for these two molecules, C1q and NC3, because there are known variants that affect other kinds of human disease. Not so much brain and spinal cord like central nervous system, neurological disease and injury, but known variants that have an impact on other diseases of the peripheral systems. I'm incredibly interested in that. And we haven't had the bandwidth. I don't know the answer. But it's a fantastic question because you would predict that that's going to have an influence. Absolutely.
So basically, this is on the roadmap. It just doesn't have a date yet.
Exactly.
I would say that most importantly, should we ever get to do this in people, we always build those clinical trials with a lot of biomarkers. So I wouldn't be excluding, I mean, I would be 100% sure that part of those potential clinical trials will include measuring the expression of the receptor on the tumor and trying to correlate to the way that the patients respond to the treatment. Because we have learned from other antigens that the more expression you have, and more important, the more cells with the expression you have as percentage in the tumor, because no tumor is going to have 100% expression, the better the response of the patient might be.
Oh, sorry, there's one. I'll come back to you. You can be our closer.
I was curious for the treatment that's currently in clinical trials involving the tumor resection and then training your T cells to recognize the antigens, if you guys had considered adding radiation to the mix. So I had recently seen a poster from Cornell that did a combination of irradiating the GBM and then doing combination immune checkpoint inhibitors, which greatly increased efficacy.
So great question. First of all, that was a newly diagnosed patient study. So by default, all the patients received radiation immediately after the resection. One thing that we haven't discussed here is that making cellular therapies, it's very different than taking a drug off the shelf. There is an innate time that it takes in order to purify those cells, grow those cells, condition those cells. Very similar if you do CAR-Ts, very similar if you do NKs. Those personalized treatments have to be processed. So while we were doing the process, the patients were receiving the radiation. And there are lots of data suggesting that maybe the radiation itself, by destroying some of the cells, increases the immune response. But that's something that we still have to fully understand, because you are radiating the niche. And while you are radiating the niche, destroying cells, getting responses in the circulation, you also are changing the immune systems in the niche. So we have to understand much more when is the right timing of getting the immune treatment after radiation.
So you guys mentioned in your slides a bunch of terrible diseases that you guys are trying to treat or cure, like neurological diseases and stuff like that. And I was wondering if you guys also do research in maybe things that could enhance humans, such as maybe like making people more intelligent or increasing longevity, sort of like Elon Musk, like Neuralink.
Well, there are some ethical issues with that. So no, we do a lot of different kinds of research in the center here. I suppose the closest you would come in the regenerative medicine sphere would be, there's a really active area of interest, not so much on this campus, a few people that dabble in longevity, right? And whether it's possible, I told you I had no cough, and here it comes. And whether it's possible to, extend health span, if not lifespan, by virtue of some interventions. But to actually try and make someone smarter or engineer a better human, that's not a thing that is so much done.
And again, the ethics of that are questionable. What I will say that, by example, as you know, right now you can do gene therapy to create people that are resistant to HIV. That's the way that we can potentially cure HIV at this point in time. We can take bone marrow stem cells. We can do CRISPR changes to them to exclude the receptor for the HIV intrusion in the T cells. Then you can transplant the bone marrow back, and those patients, even if they have HIV, the HIV cannot go in the immune system, so they get physically cured. So lo and behold,
So
this...
that's very true, right? And in CRISPR-Cas9
Yes.
gene editing studies, very much in the regenerative space, things that are being done by investigators to treat not just HIV, but severe combined immunodeficiency, the alpha thalassemias, but not, for example, to create a better human in the sense of engineering an embryo. right?
However, somebody has done that,
Yes.
and
but
that is very
with
unethical.
a lot of condemnation.
And a lot of condemnation when... Yes, he is in.
Yeah, I think pretty much
he's
disappeared.
somewhere, but not anywhere where we like to know. So, manufacturing babies that are HIV resistant in vitro, it's something that I would not encourage or condone.
Pretty much any reputable scientist. So. With that, Daniela and I will be here for a few minutes. Thank you very much for your attention and for coming tonight, and happy voting.